I have to laugh, first of all, thanks to Dr. Friedson for the introduction. I have to laugh because the last time I think she introduced me for something was at New England ACSM at the Student Investigator Award session, and that was back during, I'm old, they're back during the slides, and so they had them in a carousel, and the person in the back dropped the carousel, so the slides all fell out, and Patty and Priscilla just made a beeline for the back of the room to try to put everything back together again. So I just had a flashback to that, but we'll go for that. The introduction was odd. I sent Patty my biosketch, and so if you know me as an obesity nanoparticle person, which is sort of what my lab does right now, ignore that. I am a muscle damage person because I'm going to talk about muscle damage today. And I have been a muscle damage person since the mid-1990s when I was at Texas A&M University working with Sue Bloomfield and Bob Armstrong on an animal model. So this is, my history with muscle damage goes way back. Those of you who sort of have a first love in research, exercise-induced muscle damage is my first love, and I play in it as many times as I can, even though my field of research has gone off a little bit sideways. So I want to make everybody participate for a second. On program committee, we're always talking about making people exercise. So for a second, muscle injury. How many of you guys have experienced this? I play a lot of contact sports. Bruises, either real or imagined tears in the muscle, depending on if you're playing English Premier League soccer. This happens to me all the time. I play a lot of contact sports, but this is not the muscle injury that we're talking about today. The muscle injury that we're talking about today is this type of injury. And so how many of you guys have experienced this? And I want everybody's going to raise their hand. Everybody in here has been sore. We all get off the couch from time to time and try to do something that we're not used to doing. And so when I talk about exercise-induced muscle damage, I'm talking about that delayed onset muscle soreness, this sort of sustained weakness that you get after you do something, typically when it's novel. And so given that, we're going to start off with what my idea was to give this talk. And I'm very appreciative to the committee that selected me to give this talk. It's a huge honor. Priscilla made more of an impact on my life than anybody else, both professionally and personally. She made me who I am today. And so I'm happy to show you the type of work that we did together. And so she gave the Wolf Lecture in 2007. I'm sure you all just came from Dr. Kramer's Wolf Lecture. It's really odd to do your dissertation right before your professor gives the Wolf Lecture, because you get to see your data on the screen. And I'm going to show you a slide today that's the data from my dissertation that made it into that talk. But Dr. Clarkson gave a fantastic Wolf Lecture about muscle soreness, causes, consequences, and cure. So we'll talk a little bit about soreness today, but mainly today we're going to talk about what's going on in injured muscle. Let me start my clock here so I don't go over time. So my idea for the lecture was, let me tell you what happens after the wolf. And that was sort of my preliminary title. What happens after the wolf? Dr. Kramer is at the top of his profession, but he's not stopping today. The stuff that he showed you today is going to go on for the next 10 years, 15 years. And she did 10 years of research after that lecture that I was involved in most of it. So what I'm going to show you today is sort of what happened in the next 10 years. And that was an exciting 10 years. I'm going to step back and give you a little bit of a history lesson all in one slide. Exercise-induced muscle damage really started, it's been happening to us since we were, cavemen, but the first report from 1902, and you see this slide that's in gray, a little bit different background than what I have. I stole this from the Wolf Lecture from Dr. Clarkson. She stole my slide, so I figured I could steal hers back. And so basically this monograph by Theodore Huff in 1902 started out this idea that exercise-induced muscle damage was caused by micro tears in the muscle. But in 1902, there was no way to prove that. So he had suggested that soreness has its origins either in some type of rupture within the muscle or of the connective tissue. And that's a key aspect because we're going to talk a lot about connective tissue today. So after 1902, there was a bit of a delay in trying to figure out how to prove this theory because we didn't have the technology to prove that there's micro tears in the muscle. And this is a bit of an inside joke. So we had a couple of world wars, but then the 60s, I TA'd for Dr. Clarkson for 5 years, so 10 times I saw her try to tell the same joke about free radicals, and the punchline was Abby Hoffman. If you're doing this to a crowd of 218-year-olds in a sports nutrition class, you get crickets every time. So she was one of the funniest person I've ever met in my life, but she could not tell a joke. She tried to tell this joke about Abby Hoffman 10 times, I saw it fail 10 times, which was funny in itself. So really in the late 70s and early 80s, we gained the ability to actually see inside the muscle at the electron microscopy level to prove that there were indeed micro tears in the muscle fiber. And in the 1980s, there was a lot of work being done in different labs, including Dr. Clarkson's, including Dr. Armstrong's, to really provide these lab models so that we could test different hypotheses related to this phenomenon. Because if you go out and get sore, you're all doing different things. So I can't really tell what you're doing versus what you're doing to be able to tell what's actually going on. The 1990s came along and we had established the lab model. So at that point, people were talking more about variability. So Dr. Kramer mentioned this. I think he called me out and said I'd be talking about the genetics of this, which I am, I promise you. But variability is basically you walk into a gym and you get super sore from the same exercise as somebody else walks into the gym and they don't get as sore or they don't get as fatigued. So there's this variability in this response. There's also this idea of adaptation, and this is the coolest thing ever, because this is how resistance training causes hypertrophy, causes adaptation. It's at the molecular level. How is the muscle changing so that it is stronger and bigger or whatever you want it to be with training? So in the 1990s, Dr. Clarkson and others showed this adaptation to exercise-induced muscle damage and generated more indirect markers that they could see with regard to how much somebody responds to exercise. What I'm going to talk about today is the last 10 years or so. So starting in the early 2000s, revolution, and that revolution is caused by 3 words: Human Genome Project. So I ended up in the Clarkson lab in 1999. Human Genome Project just started, and my second year there, gene chips came on the market, and I switched my entire dissertation around from very functional to very molecular, and it was quite a ride from the beginning. So in the 2000s, really people started looking at this from a molecular biology and genetics fashion in healthy muscle and also in things like dystrophic muscle and neuromuscular disease. And in the 2010s, Human Genome Project kind of settled down, so now we know all the players in the game, but we have no idea how they interact with each other. So I'm in a systems biology department. That's what we do. We take these complex systems and try to understand them. And you saw some of the webs that Dr. Kramer put up. Dr. Hill put up a web last night of obesity. I thought I was the only one showing complex stuff. But so in this slide, what I'm trying to do is show you the evolution of how you look at something from a molecular point of view. So I'm doing it with muscle damage, but it's obesity, any other sort of thing that we're looking at molecularly. We use a lot of gene chips, and this is a transcription global process. You can look at all the genes at one time, what's going on. And what this is telling me, sorry for the people in the back if you can't see this, the key question is, what is a damaged muscle cell thinking? So what is it thinking? How is it insulted? How is it responding to this insult? And so in 2003, we published our first study, and I think it was the first muscle damage molecular study using gene chips. And this was the state-of-the-art bioinformatics in 2002, 2003. We basically took everything that went up 500% and tried to summarize it using a category. And I came up with the analogy of sort of doing a census. If you walk into a town to do a census and the first 20 people come up to you, you say, okay, 3 of them were named Bob, 2 of them were named Mary. You have these general descriptions where you don't really kind of know a lot about them. So we thought, and this represents 27 genes out of about 25,000 genes. So these are the ones that went up gangbusters. I thought I was the only one that's going to show a scary spider web. Don't worry, you don't have to understand the spider web. You just have to understand that instead of 27 genes now, we're actually describing over 1,500 genes using these maps to sort of figure out who's related to who. So in the census analogy, you go into a town and you meet 1,000 people, figure out who's in what family, who's doing what job, who's interacting with who married, between certain families. And what I want to point out is, it seems like there's not many more than 27 things here, but each of these boxes is actually 35 genes. So there's more genes in one of these boxes than there is in this entire pie chart. So you're going to see these a couple of times, but in the end, what I hope to do is tell you what this is telling us. So I'm going to spend the entire talk sort of building you up to that. So, lecture outline is this. I'm going to give you a short primer on exercise-induced muscle damage. Some of you are students, some of you work in other areas. You don't have any idea what exercise-induced muscle damage looks like. We already ascertained, you all know what it feels like. You don't know what it looks like. And so we know very well what it looks like and how it's sort of manipulated by different things. So I'm going to get everybody up to speed on that. Then I'm going to tell you 2 short stories. I, like Dr. Kramer, can't tell you everything about resistance training in an hour. I certainly can't tell you everything about molecular muscle damage in 40 minutes now. So I'm going to tell you 2 stories. One story is about structure. So how does the structure of the muscle change with damage? And especially we're going to talk about the extracellular matrix. The second story I'm going to tell you is an inflammatory signal, and these are not independent stories. They're mixed. They interact with each other. But I'm going to tell you one each sort of vignette, and then I'm going to end with what causes variability in muscle damage. So just to get everybody on the same page. What do we do to cause damage? We typically use eccentric exercise. And if you're in the gym, this is what you think of as lowering weights or negatives. And the reason that we use eccentric exercise, and the eccentric exercise is when the movement is lengthening. So you have a big weight in your hand, it's more than you can curl, so it's going down. And these are the negatives that you would do in the gym. And you know that in the gym with the negatives, you can do higher weights than you can do with a positive. I can lower down a weight heavier than I can push up. So those of you who haven't taken biomechanics before, I'm going to give you one clue about biomechanics, and that's the force-length relationship of muscle. So the maximum force on the y-axis here versus the length of the muscle proportionate to resting length. So 1 here is the resting length of your muscle. As you expand that out, you see the total force peaking a little bit above resting length, and then coming down, and then it goes up again. And why does it go up again? Because the active force is going down. The ability to contract is going down, but the passive forces are going up. So you're loading structures that aren't providing active force, they're providing passive, don't tear me apart force. Okay, and that's bad. You don't want that, and that's what's causing the damage. So in the Clarkson lab, she developed this great model where we could produce a moderate amount of muscle damage, and this is key, moderate amount of muscle damage with no clinical complications. You're not sending anybody to the hospital. You're not, not having anybody come, be months later and still impaired. So we've got Ted here on a preacher curl bench who's trying to pull his arm up towards his face. Okay, he's trying to do a preacher curl. There's an enthusiastic grad student on the other end of this pole. They've got the mechanical advantage. They're going to win every time, kind of like playing the slot machine. So you do this 50 times, 5 sets of 10, or some variation of that. You're going to make somebody's arm very sore. Couple advantages to using the arm are that it's a small muscle mass, so you're not going to create a large amount of damage. It's an expandable compartment, so it can swell like the dickens. You can see people come in with bowling ball arms, and there's no compartment there, so there's no compartment syndrome or anything like that. So my time in the Clarkson lab, we did over 1,000 subjects. We used to do these large clinical trials for pain medication. What you don't want is this. You don't want exertional rhabdomyolysis, which is extreme muscle breakdown. Hallmark symptom of that is Coca-Cola colored urine. So myoglobin gets out of the muscle through the muscle tears, gets into circulation, kidney has to clear out all that protein, some of it spills over into the urine, you get dark urine. Never in the Clarkson lab did we have anybody who had clinically relevant rhabdomyolysis, even though we exercised well over 1,000 people. So this nice, safe lab model. So to bring now that I showed you how we incur damage, let me show you what the damage looks like. So everybody knows this. Starting with your muscle all the way down, we're going to start at muscle at the sarcomeric level. So if you bring it all the way down to the size of the fiber, you have the sarcomere Z-line to Z-line with the actin and myosin in between. So actin is anchoring to the Z-line. And again, in the 80s, we had the ability to see what normal muscle looked like. And then in the 90s, we were late 80s, early 90s, we were able to see, again, this is what normal muscle looks like. This is what 2 days after an eccentric protocol looks like. 2 things that are hallmarks of what I'm going to call damage. And if there's anybody from Europe in here that wants to really throw down and argue about whether this is remodeling or damage, I'll meet you in the hallway afterwards, because I'm saying it's a semantic difference. It is remodeling. You have to do some damage before you fix stuff. Use the analogy of remodeling your bathroom. You're going to do some, it's going to look worse before it looks better. But I'm going to call it damage for the purposes of the talk. You see 2 things here that are hallmarks of damage. Number 1, sarcomeres that have just given up. They've been stretched beyond repair. They're going to get edited out. Other thing is Z-lines are no longer lined up. They're no longer in registry. Okay, so that's what damage looks like. We've all have seminal moments when we see data and we think that's the greatest thing ever. That's coming up for me. Greatest thing ever, and my alternative title to my dissertation is Exercise-Induced Muscle Damage. If you do nothing, it'll fix itself. So in healthy muscle, If you leave it alone, it will fix itself over time. So you see here, it's not completely normal, but it's back to looking fairly normal, but it's remodeled. So what happens is, if you do the whole thing again, you get what's called the repeated bout effect. It said, I saw that, I'm suspicious you're going to do that again, so I'm going to remodel so that that doesn't happen to me again. So the repeated bout effect is something Dr. Clarkson was best known for in the 80s. The idea being performance of one bout of exercise essentially makes you adapt so that you're resistant to the next bout of exercise. This is what training is, people. You go in the first time, you're super sore, it affects you a lot. After that, not so much. So the way to fix exercise-induced muscle damage, unfortunately, is to do it again. So indirect damage markers, I like to do biopsies. I like to poke people with needles and get muscle biopsies out, but lots of people don't like to do that. It's expensive and you need biopsy needles. So a lot of studies are done using indirect markers: force loss, soreness, muscle proteins in the blood, and inflammation. Due to time constraints, I kind of put them all on the same slide, except for inflammation, because we're going to talk about inflammation. So strength loss, and this is what typically happens after that TED exercise with the 50 maximal contractions, or if you do it in the leg. You get a drop in strength immediately afterwards, and here's the most important thing to understand. You get a second drop in the hours after exercise. So if this was metabolic fatigue, you would see it go back up as soon as you've rested. Soreness is not lactic acid, people, okay? It's not metabolic fatigue. What you see in this first part of the curve is mechanical damage and a little bit of fatigue. What you see in the second part of the curve is a secondary damage caused by inflammation. So that's why we're going to talk about inflammation. Then once you get to 24 hours, you start to recover so that 5 days later, you're back to normal. Not statistically different from baseline. This is highly variable. Some people lose less than 10% of strength, same model. Some people lose 90% of strength. There are some high responders that take a long time to recover. There are some people that recover within a day or two. So each of these is highly variable. So in each case, I've shown you the mean response, so about halfway up the soreness curve, peaking at 48 hours, coming down at 4 and 5 days. Lots of variability. Some people ranking it on a score of 1 to 100 or 0 to 105. Other people 95 or 99. So lots of individual variability. This is creatine kinase activity, stuff that's supposed to be in muscle that gets outside of muscle, goes up, highly variable. A lot of these variability error bars are small, because this is 200 people in a trial. But you see the individual variability. So here you see lots of people with 0 to 10,000, 0 to 1,000 CKs. You see other people at 30,000, 40,000. Thanks to Ken Nosaka for these data, because I put them up next to each other. If you do that second bout, again, you've adapted, so all of these markers go down. You see, this primary, damage, the secondary damage. You see the primary, decrement after the second bout, which is in the open squares here, but then it comes up faster. So not as much damage, not a lot of secondary damage, and faster recovery. Soreness, goes up a little bit, comes down faster, and this is what's really interesting. CK is almost non-existent after a second round of exercise. We're going to talk about why this is. It's basically remodeling of the extracellular matrix and the membrane. Inflammation. So the generic inflammation, you see people with giant bowling ball arms, swollen elbows. You see, if you just measure circumference, it goes up, 20 millimeter bigger arm. If you look by MRI, you see lots of swelling in the bicep because they're doing bicep exercise, nothing in the tricep. This is the same cross section. But here's where we start to get a little more complex. So if we try to look at inflammation not just using circumference, but trying to figure out what's actually going on in inflammation, we have to go into the inflammatory literature, which is very complex. So if you love this stuff and want to know more about inflammation, any of this stuff, I have several review papers that I can suggest to you. You can email me. This is adapted from Jim Tidball's review. He has several on the topic. What I want to point out is that you can sort of summarize the inflammatory responses biphasic or triphasic, depending on whether you're talking about destruction versus regeneration or what's actually going on with the cells. So going back to my bathroom remodel analogy, you have a cousin who's strong who likes a sledgehammer. They are this destructive phase. They're going to be helpful because they're going to do a lot of destruction pretty quickly, but you can't sort of let them get out of control because then you got to bring somebody in who knows how to put stuff back together again. So here we have a destructive inflammatory, pro-inflammatory response. It's coming in to fix the damage, but it goes a little overboard in sort of trying to remodel. Then you get this regenerative phase that comes in and puts everything back together again. So what is part of each one? The destructive is two things. Number one, neutrophils. Neutrophils get activated and they put out a lot of oxidative radicals. Then you get this sort of secondary, but still early, about 24 hours, monocyte invasion, lots of macrophages, and these are pro-inflammatory macrophages. And then the whole thing kind of switches over where the inflammatory cells that are dominating are more of the anti-inflammatory type. So we're going to revisit this in a minute. So to summarize the primer, we basically think of muscle damage as two things. Number one, you've mechanically torn something apart. So you have damage, you've got to fix that. Then you get this secondary damage where you got to fix what's there, but you also got to sort of remodel and set it up to be regenerated. One of the things that's really interesting about this is that if you try to mess with one of these, you mess with the other one. And the hallmark evidence for this is NSAID use. So if you use nonsteroidal anti-inflammatories, you can drop your soreness levels. However, what happens is it decreases all of inflammation, so your regenerative response is actually attenuated. So it actually takes you longer to recover. So what are you going to do? Are you going to pay the price early, or are you going to pay the price late? So you can't really mess with the system without knowing what you're doing. So what happens actually inside the muscle? There's a lot of data out there about what happens to produce this force loss that occurs and persists for several days. Rob Hildahl and I, mainly Rob, did a review paper in Muscle and Nerve in 2013. If you're interested in this stuff, I would highly suggest reading that paper. I'm going to show you all of the different things that have data for them, but I'm only going to talk about one of them further. So this is 5 different things that can cause force loss. And remember, this is force loss over several days. It's not metabolic fatigue. So you have number 1, so the sarcomeres that have sort of given up. You don't have as many cross bridges to link together, so your strength is lower. Number 2, the stretch channels in the membrane may be disrupted, so you don't get the good calcium signaling that normal muscle has. Number 3 is breaks in the membrane, and I added in the ECM part. It's not in that paper, but I added it in because that's what we're going to talk about today. So if you break the membrane or the ECM that sits on top of this providing structure, you have things getting in that shouldn't get in, you have things getting out that shouldn't get out, and that causes a problem. So we're going to talk a lot about that in the next story. 4 is excitation contraction uncoupling. Bob Armstrong's lab and Gordon Warren and Chris Ingalls have done a lot of work on this type of thing in the days after exercise. Basically, this whole system where you depolarize and cause contraction gets messed up for several days. And that's not the technical term for it, and I have several reviews that I can suggest if you want to know more about excitation contraction uncoupling. And then there's this idea of degraded proteins, so calpains and other things that are activated by the oxidative stress can break down proteins that it's not supposed to. So we're going to focus on this for our first story. So our first story, again, extracellular matrix changes, relationship to dysfunction and adaptation. I told you this is the first thing that we did, and it was an N of 3, and we didn't know if it was going to work, and it got published with an N of 3 because it was 2003. Not a chance in heck that you could get this published today with rudimentary bioinformatics, but it turns out we were right. So the first story I'm going to tell you is about these structural genes. So the extracellular matrix, if you don't know much about it, it sits outside the muscle membrane, but it really communicates with the muscle membrane and provides, contributes to force generation. It's typically divided into like epimysium, paramysium, and endomysium. There's some argument about whether those are distinct categories or not, but you see the microscope, electron microscopy structure here. You see the stain structure here. And Rick Lieber's group and others have shown that if you look at bundles that have the extracellular matrix versus individual fibers, you actually get higher amounts of force ability. So the ECM is definitely contributing to that force and that passive force that I showed you in the force length relationship. Maria Urso gave me this. I have a way too simple slide and a way too complex slide, so you're going to have to average out the two in your brain. do a little bit of work as an audience to figure out how much you want to know about this. This is the too simplistic version of this. You have a cell, this is a generic cell, so I know muscle cells have multiple nuclei. This is a generic cell sitting on its extracellular matrix, and it's bound by proteins. So proteins go across the border here. If you stretch out the extracellular matrix, the cell is going to sense that from these proteins and send signals to the nuclei to do something about it. I'm sensing something, I need to do something about it. It's raining, I should probably get out an umbrella. Like, something needs to happen in response to this. So the extracellular matrix provides force, but it also provides the stimulus called mechanotransduction, and it happens in bone, it happens in other tissues, it happens in epithelial wound cells, or cells that have been wounded. In muscle, it's key. This is the way too complex slide. So this is a slide from 2013. We know a lot about the extracellular matrix and how it reacts with the membrane. So I don't want you to understand all of this. What I want you to understand is that there's so many ways for it to communicate with the muscle cell. So you have different cytokines and different hormones and different growth factors. You have membrane or extracellular matrix proteins that talk to inflammatory proteins or growth factors, and these transmit through the membrane through some of the intracellular intermembrane proteins like dystrophin complex, like the integrins, like TGF-beta receptor, to cause gene expression changes. And lots of these gene expression changes are what we think of as stress responsive. Lots of them are what we call inflammatory. So this is how the two stories sort of overlap. So you don't have to know what each of these does, just understand that there's a really complex relationship between these two things that are sort of married to one another. So Rob Hildahl and his group, including myself, just had this accepted into the FASB journal, so you can look for this coming out soon. What Rob did was he took a existing study, one of the last studies that Dr. Clarkson did. We went from 3 studies in that 3 subjects in that first study to 35. So this is a really big microarray study with 1 bout of exercise, 3 hours after exercise, biopsied leg versus control leg. What is the muscle cell thinking? What is gene expression going on? So Rob put a heat map up here where everything you see in green goes up with eccentric exercise. Everything in red here is going down. So you see things are going up and things are going down. It's never all in one direction, especially because things are complex. So you have TGF-beta and tennescin and some other things that are going up, including the integrins that are in that membrane. You have a bunch of the collagens going down, and this is actually really important when you think about having to put stuff back together again. First, what you have to do is sort of take apart the structure that you have to be able to put it back together again. So Malachi McHugh has had several reviews put out where he's tried to sort of get to adaptation from a 50,000-foot view. So if you sort of think about these grand hypotheses of how muscle adapts, some of the suggestions are that it's neural adaptation, which we sort of don't believe so much because it can happen even if you take the brain out of the equation. There's tissue-level adaptation with altering the ECM and the membrane and the stiffness properties of it. So this is that first story that I'm telling. We have sarcomere-level adaptation where those weak sarcomeres sort of get weeded out. And then the systemic adaptation through altered inflammation. That's the second story I'm going to tell. So I'm taking a little bit of a side thing to just sort of orient you to the extracellular matrix and inflammation play big roles in adaptation. So this is the same slide that I showed you before, and I'm highlighting these 2 things, laminin C2 and integrin alpha 7, because I'm going to show you some more data for this. So remember, this is 1 bout of exercise in a lot of people, so the data are really strong for this group. So what Rob did was he took a new group of people, exercised them 1 time to get the 3-hour data, and then he exercised them again a month later, and that's within the window of adaptation. So what happened is in laminin C2 and in integrin alpha 7, you see here's baseline levels, here's the first bout of exercise. You see a large increase. This one's statistically significant, this one's not. I hesitate to say it's not significant because I just want to show you the raw data with the error. It's highly variable, but you see the data. So it probably is going up here. See it going back to baseline, and then you see this attenuated response the second time. And that's what you see with strength and soreness and all those other things. You see this attenuation of damage the second time around. And you see it in laminin C2, which is part of the extracellular matrix, and you see it in integrin alpha 7, which is part of the membrane. membrane. So again, increase back to normal, increase but slightly less so. In fact, it's not statistically significant anymore. If I went back to the previous slide and showed you, Rob has data from several of the other collagens, the other integrins, they all don't show that exact same pattern. This is what we call the classical repeated bout pattern. Some of them go up a little bit after the first bout, and then go up a little bit later, and then go up again after the second bout. So it's not all the same pattern. I showed you these two, they have the classical response, but it's really complex as to how it's put together. So kudos to Rob. He went back to a Mackey paper from 2011 and started there and then added these data on top of it. So I want to sort of provide this theoretical model of what's going on. So you don't need to know the individual genes and sort of what they're doing or what proteins are being made. Here's what's happening. You have the extracellular matrix sitting outside the muscle fiber. And what you see at first is this idea of de-adhesion. So basically sort of, taking the structure that's together and sort of loosening up everything, sort of, de-adhesing everything, sort of, allow for something to come in and sort of restructure. So in this phase, which is early, hours and 1 or 2 days afterwards, you get force loss, membrane's permeable because it has breaks. Senescence C, which is an indicator of the membrane, is up. Integrins are up, so that pieces of the membrane are they have higher gene expression, so they're really trying to do more to put the membrane back together again. You get activation of satellite cells, and for those of you who don't know what satellite cells, we'll get back to them in a second. But basically, all these things are going on to sort of loosen up the structure. Then there's restructuring. So now, okay, the alarm is over. We've got some time. We've got some energy. Let's put this thing back together again so that it doesn't happen again. All your body cares about is, let's not let that happen again. We were kind of unhappy about that whole situation. It was painful. We lost function for a while. So during the restructuring phase, you get more TGF-beta signaling, which is more growth factor signaling, also overlaps with inflammatory signaling. The collagens start to express more, so you're putting back together the extracellular matrix. You've got satellite cell content going up. Again, we'll get back to satellite cells in a second. But here's what's really cool. So what's happening in the repeated bout? So what happens after you've put stuff back together again and you do the whole thing over again? You don't get everything being torn apart as much as it was the first time. You get a more subtle signal to say, hey, this is happening again. So we made the big changes the first time around. You're remodeling your house. You put on a new floor. Great. Next time around, you're like, maybe I'll do the bathroom next time. So we've already done the big project. Let's go for the little projects and sort of make it perfect. So in the repeated bout, you don't have as much force loss or membrane permeability. Satellite cell content is unchanged. Senescence C, again, membrane breakage goes down. More growth factor signaling. So hey, we still need the growth factor signaling. Collagen expression depends on the collagen. Some go up, some go down, some don't change. Integrin expression is generally down. So that first phase of sort of restructuring the membrane itself and those proteins is somewhat downregulated after the first bout. Because you fixed the extracellular matrix, maybe that much force hasn't transmitted into the muscle fiber after that. So that might be all part of this better, stronger muscle the second time around. So that's the first story, and that's a structural story. It's a little bit of an early phase, like fixing the mechanical damage story. It's a little bit of the sort of later, what happens with the inflammatory phase and comes in and it all sort of puts itself back together again. Now let's talk about inflammation itself, because again, can we affect this secondary damage? We can't really avoid, unless we want to avoid anything that requires us to move our muscles in a weird way. You can't really mess with the fact that you're going to get mechanical damage, but what you can mess with is this inflammatory response. So in my earlier analogy of your cousin with the sledgehammer coming in, you can sort of talk to him beforehand and be like, don't go crazy, or sort of get him out of the way sooner rather than later and mess with that. But again, it's the butterfly effect. If you mess with the front end, it's a very complex process. You might not get the signals for the back end. So let's talk about inflammation signaling. Again, stolen from, probably with permission at some point in my life, stolen from the Wolf Lecture. So this is a Wolf Lecture. And this is how Dr. Clarkson married the two together. So they're not two distinct stories. It's all part of the same process. What she had suggested was that damage sounds the alarm. So this extracellular matrix damage sounds the alarm through mechanotransduction to the muscle cells to start doing stuff, start responding. And it does that through gene expression. And this was in 2007, we were still explaining to people about gene expression and how genes make proteins. So I hope everybody remembers their DNA, RNA, protein. Didn't have time to put that into this talk. But basically, genes provide the instructions to make proteins. So now you're making proteins. Some of those proteins that are responsive to that damage alarm are inflammatory. And again, that's what causes secondary damage, but that's also what causes remodeling and adaptation. So can we mess with that? So again, we were somewhat lucky that the first time around, using rudimentary bioinformatics, we had a couple of targets in inflammation and in cell cycling, cell regulation, because a lot of these are inflammatory. Let's go back and sort of look at these a little bit more. This is sort of the middle ground between the really simple pie chart and the really complex spider web that I showed you. What we're looking for when we look at 20,000 things at the same time is everybody doing the same thing. And somebody explained this to me one time as you're Napoleon standing on the hill at Waterloo trying to figure out what the army below you is doing. One guy goes to the back, and he said it's really hard to use microarrays to detect a flanking movement in one individual. So one guy starts moving to the left. You don't know what he's could be going to the bathroom. He could be going to pick up his sword. But if a whole troop moves to the left, something's going on. So what we do in high throughput data is what family is going in this direction. So what Rob did, again, this is from the sports study, so 35 people, 3 hours after exercise. He found 36 different genes that were changing. And what does this all have in common? I hope you can see this. All points to nuclear factor kappa beta. So a lot of you know what NFKB is. It's a key growth factor signaling network. It's also an inflammatory signaling network. So it does a bunch of stuff in the nucleus. It has impact, so things in the cytoplasm, plasma membrane, extracellular space, including the extracellular matrix, all sort of signal down to NFKB. Why isn't NFKB colored here? Because the key signaling pathways, the key members, AKT1, NFKB, TNF-alpha, if you know any of the key pathway stuff, never changes transcriptionally. And the body's not designed to do that. You're not designed to wildly turn this on or wildly turn this off. So what these do is these change at the protein level. They're activated or repressed using phosphorylation or other protein signaling. So what this said was we need to look at NFKB at the protein level. So Rob did that. And again, this is again from the sports study. It's an earlier study than the repeated bout study I'm going to tell you about. I had them dim the light, so I hope you can see this a little bit. Here, whenever you see green, that's NFKB. And in fact, for the aficionados, it's the P65 subunit of NFKB. So it's in green. And you can see in the control muscle, this is a cross-section, so this is a muscle fiber. And you can see the nuclei are stained in red here. In green, you see NFKB, but you don't see a lot of it over here. This is after eccentric exercise. You see lots of fluorescence over here. And in fact, statistically significant increase in NFKB after eccentric exercise. But what's really interesting is where it's happening. Here's a muscle cell. It's not happening inside the muscle cell, it's happening between the muscle cells. That's where the extracellular matrix is, but that's also where capillaries are, inflammatory cells are. There's a lot going on in there. So we took this picture next door. My office is one place. Next door to me is a guy named Terry Partridge, who knows more about satellite cells than anybody else in the world. And I said, "What's this?" And he put it in and popped it in his computer, and he came back and said, "That looks like a parasite." And I waited for a second and I go, "What's a parasite?" I'd seen a lot of these images. I'd worked in the field for 8 or 9 years at this point. I knew what a macrophage looked like. I knew what a satellite cell looked like. This is a parasite. Parasites are mesenchymal stem cells that surround endothelial cells. So in fact, parasites are specialized forms of endothelial cells that surround the vasculature. So you're messing with the capillary bed that's inside the muscle. So you're not only tearing the extracellular membrane, extracellular matrix and the muscle membrane, but also messing with the vasculature. There are others of these things that are co-localizing, where you see yellow here, it's a nuclei that's expressing NFKB. You see over here on the right, eccentric exercise versus control in the myonuclei doesn't change, but in the non-myonuclei it goes up. So non-myonuclei in this case, parasites, satellite cells, macrophages, other things. And remember, when we take a punch biopsy of a muscle, you're not only getting the muscle cells, you're getting the parasites and the capillaries and all this thing. You homogenize it all up together. So the signaling is happening in a very specific place. So let's talk about what the role of satellite cells, and we're going to include macrophages here. Basically, this is a Tidball review from 2005, but I love it because it really sort of sets the tone for intracellular communication. What you have here is a neutrophil sort of driving towards injury via muscle lysis. If he was doing this paper today, these would be labeled M1 macrophages, or those pro-inflammatory macrophages. On this side, these would be M2 macrophages, and these are using chemokines and other chemicals to signal between the satellite cells and the macrophages to affect change. And in fact, what you're trying to affect with the satellite cells is for them to come fix stuff. So this is a really, early at this point, 2001, review by Tom Hawk, where you see a resting myofiber where you get myotrauma, and again, that mechanotransduction comes in and basically causes the satellite cells to become non-quiescent. They activate, they proliferate, some of them go and fix stuff, some of them go back into replenishing the pool. So these proliferate, some of them go back to the pool, others go to injured fiber, and this word is key, chemotaxis. What causes chemotaxis? It's a lot of these chemicals. Fusion to the damaged fiber, some of them make new myofibers, and then you have regenerated myofibers that then mature. So you need this to regenerate fibers. So we looked at some of those from 2 slides ago. Some of these things, again, this was 2005. We knew a couple of things that sort of helped these talk to each other. Based on the last slide, NFKB helps them talk to each other. And I created some data when I was still at UMass, my final dissertation study, where I looked at a bunch of inflammatory genes. I told you that inflammation in general, CKs go down, attenuated inflammation after the second bout because you've adapted. So I figured I would look at these inflammatory genes and they would go up like gangbusters after the first one and then show that attenuated response after the second one. To my surprise, I tested 8 using PCR and a good sized number of people. Turned out 5 of them didn't do anything different. They went up gangbusters after the first time, same thing after the second time, no decrease. 3 of them went up even further. So I don't know how many of you have had a hypothesis that became completely the opposite of what the data showed. My final dissertation study became, hey, something's going on that's increasing parts of inflammation, and it happens to be one of those genes is called MCP1, monocyte chemoattractant protein 1. The gene codes for it is CCL2. So MCP1 and these 2 other transcription factors that are 1 or 2 degrees of Kevin and Bacon away from MCP1 are basically all going up after the second bout. So if you go back to the Tidball review of what's causing damage versus what's causing, driving renewal and differentiation and good repair, these are the ones that are causing good repair. So now you've primed the system to be better at repair. So that was really cool. I think a lot of this stuff is really cool. Hope you do too. So we localized MCP1 protein. So where is it? Here's a control leg, and I had them turn the lights down, but this is again a cross-section. So here you can see the cross-section by the nuclei sort of ringing the structure. This is the eccentric leg, and MCP1 here is green. So you see lots of MCP1 after the second bout. So again, goes up like gangbusters after the first bout by 5-fold, then goes up again another 2-fold beyond that, so 10-fold above normal after the second bout. And for lack of time, I'm just going to give you the punchline to the story. MCP1 is being expressed by macrophages. So here the macrophages are in red. And in here, the satellite cells are in red. So satellite cells and macrophages are both expressing MCP1. So this sort of ends the second story and moves into the third story, which isn't a story. It's more of a, what does it all mean? And I think Bill Kramer said I'd talk about genetic variability, so here it is. I didn't tell him I was going to do it, but he was right. So here's the transition. What we did with MCP1 was we thought, okay, so if this makes people repair better, if people have different levels of MCP1, maybe they're better or worse at responding. So that maybe that's what underlies the variability. So for strength loss, if you take people with different genotypes for MCP1, some make more, some make less based on how they make the protein. Anybody in here that has a C allele for their MCP1, this is one particular mutation, you're going to have a harder time recovering. And this is only one genetic mutation, one letter of your alphabet switch. So think about how many. I had a slide that I had to take out for time, but there are probably 24 different stories now with a whole list of inflammatory genes that have mutations, structural genes that have mutations, including titin, including MCP1, including the integrins, that affect variability. And in fact, MCP1 affects variability in strength, loss, CK, and soreness. So this is the last part of the talk, variability in exercise-induced muscle damage. And in fact, this takes off of Priscilla's last slide at the Wolf Lecture. I believe that understanding the causes, consequences, and cure for muscle soreness lies in the understanding the variability in response to exercise. So variability coming from gender, maybe, coming from age, maybe, training, definitely. You do it once, you get damaged, you do it again, you definitely have, will adapt. Gene expression. So this is what I'm going to talk about for the last 2 or 3 slides. We can't really get at this. We can do big studies where we injure people and sort of look at their response, but the adaptation is they have to do it again in order to figure out who's a good adapter. What I've done here is to use that sport data set. So 35 is pretty much one of the largest healthy muscle eccentric, it's probably the biggest eccentric damage data set that we have, and I used that to use correlational analyses to query all the genes and see what are the high responders doing, what are the low responders doing. So I'm showing you a correlation chart here because you map all 35 people and you see what sort of matches up to the strength loss. I also did this via clustering. I took the high responders as a group versus the low responders as a group. So I did this for strength loss, soreness, and CK using both correlation and clustering. So here we go back to the spider web, and I'm not sure, having talked for 40 minutes, if you understand this a lot better than I do. So here's what I'm going to do. I'm going to point out a couple of highlights. I don't fully understand this. I probably understand about 10% of this, so I'm going to share that part. So these are the strength, soreness, and CK clusters, high responder, low responder, and we pay $30,000 a year for this software, and it will not make anything not blue. So sorry about that. There's 3 sets of data up here, so I've highlighted them. And basically it's the top 10 networks. Again, each network is 35 genes in a family that are doing something, and in fact, these are the ones that are most highly related to CK response after one bout of exercise. These are the ones that are most related to soreness after exercise. These are the ones that are most correlated to strength loss after exercise. So that's great. You understand it now perfectly? No? Okay. So CK, I try to figure out what this network means. You go into the thing, you look around, you try to figure out, does it all point at NFKB? Does it all point at something I know? And in fact, I couldn't find anything in the CK that made sense to me. But this makes sense to me because CK in the blood has to do with clearance out of the muscle, has to do with your blood flow. I'm not finding the answer for this inside the muscle. So that kind of makes sense. Why would gene expression sort of explain all that? So for CK, I didn't really get any strong pathways. For soreness, I did. Calpains and thrombin signaling, calpains particularly, are part of that calcium-mediated proteolysis or protein breakdown in the muscle. Again, soreness has nothing to do with lactic acid. I want to say that first and last so that nobody leaves this room thinking it has to do with lactic acid. But it does have to do with irritating the nerves inside the muscle. So calpains are part of that process. Go through proteolysis, it releases some of these irritating things. The nerves inside your muscle get fairly annoyed with you and create the soreness. Strength loss was cool. So strength loss had 2 particularly strong signals. One was stress response, and those of you who know anything about cellular signaling, mTOR is a growth, anabolic, protein anabolism, also related to stress response. MAP kinase is the most generic stress response pathways that you can go through, including ERKs and MAPKs. But what was really interesting was by far the top networks associated with strength loss were energy related. So you go back to my pie chart, I actually had an energy-related piece of the pie that I never even got to. So this is really interesting because the people that are losing the most strength are turning on these energy pathways, and it may be that they're creating the most mechanical damage, that they need the most protein anabolism, they need all this energy to put stuff back together again. So, in summary, structural remodeling happens, this is our still evolving understanding. Structural remodeling changes with eccentric exercise, changes again with the repeated bout effect. What I want to highlight here is the crosstalk. So lots of different cell types talking to each other, and that's where we need to use systems biology and these networks to understand what's being communicated. Extracellular matrix, parasites, satellite cells, inflammatory cells. The key element that we've identified so far is NFKB. It's not the only one, but it's the one we have the most data for so far. Inflammation, similar story. Crosstalk, MCP1 seems to be really critical for this, but there's other elements as well. And then with variability, early strength loss correlating with energy metabolism may just be a marker of how much muscle damage is mechanical. And then soreness related to calcium signaling, CK related. I always like to end with a question mark because we don't have all the answers. So here's my acknowledgement slide. She meant the world to me. I'm honored beyond belief to give this talk. This is Clarkson Lab pre-Monica. Monica looking fairly angry to be in a picture. I wear the same shirt every year. It's pretty funny. And then post-Monica. So at least a dozen of her trainees are here today. We're a big family. I think it speaks volumes when somebody trains people that go on to do awesome things and be awesome people. So that's my talk. Thank you very much.